So it's a real pleasure to be back here in IPK. So last time I visited, it was still in the previous building, so I think that was even before Charlie came. So you had not just moved. And congratulations on all of these achievements in this past 20 years. And I wish you as much for the next 20 years. So we're going to switch gears now. So as Kyuho said, I want to keep talking about these different structural classes of proteins, but show you what happens with the eukaryotic proteins and their involvement in sexual reproduction. An alternative title of this talk could have been this, Paleontology Without Fossils, like we're tracking these very ancient evolutionary links with structural biology. So as you know, I took this from one of Steve Harrison's reviews, and what you've learned from structural studies of the viral fusion proteins for entry is that they go from the very left is a metastable state that is called the pre-fusion form to end up on the far right. What we call this trimer of hairpins in the post-fusion conformation. So the interactions with the cell could be binding to receptor or low pH in an endosome will trigger a conformational change in which there is first an extended intermediate that you have in part B, and then the protein will bend in half, and it will bring the transmembrane, the C-terminal transmembrane segment next to this fusion peptide that inserted in the target membrane, and that will force, as indicated by the arrows in C, the two membranes against each other, and it's the energy to dehydrate, the lipid head groups that sort of is counteracted by this force in the refolding. So all of the energy is stored in the folding of these proteins. So it is very different to the intracellular fusion proteins, you know, the snares and stuff, where you have, you can use ATP, the viruses have to do with what they have. So in the case of the snares, it is to recycle them that you need ATP hydrolysis, but there are some ATPases that drive fusion like elastins. So but now, so I just wanted to introduce this and to say what Q just introduced is that there are actually three structural classes of fusion proteins. The first one, class 1, was back in 1981, Don Wiley, who got the pre-fusion form of the influenza hemagglutinin represented here, which comes, is folded as a precursor, HA0, which gets cleaved into two subunits. It's a trimer, the precursor is a trimer, and the head in red and yellow colors is receptor binding, and HA2, the second subunit in bluish, is the one that will be the fusion protein. So acidic pH will destabilize the head and allow the fusion protein to undergo this conformational change that I just shown you. So then come the class 2 fusion proteins, in which they act as a heterodimer, and two proteins that are called as a polyprotein initially, and they have the fusion loop buried at the interface with the partner protein, and in the final post-fusion conformation you have the same thing, this hairpin that brings the two membranes together. And finally, class 3, like exemplified here as VSVs, it's a single protein that will change conformation and switch back. The herpes viruses have this one, and others like Togoto virus, which is also an orthomyxovirus. So except for class 1, so the structural studies have indicated that these memory fusion proteins, which I will call MFPs for this, within a class are homologous. So, and I like to stress this, that homology means that they have derived from a common ancestor by an ancestral, by the divergent evolution. So, except class 1 is a very simple motif, and it might be like two different subsets of structurally homologous proteins, but I will not go into this. So what I want to stress is that the focus here is on this class 2 fusion proteins, and they have one particular feature is that they are not standalone. You cannot just express the MFP and expect that it will function. You need to do it as a heterodimer, having the other protein that is the chaperone that will control the triggering of the conformational change to go to the postfusion form in the right. In addition, it has the property of making irregular lattices, which the other classes do not have. They are just mobile on the membrane. So here you have, in this case, icosahedral symmetry. So what I want to, something that was not really anticipated was this accompanying proteins that they turn out to be related, as you'll see. So here's for the case of flaviviruses, the accompanying protein of APSPRM. The next one is the alpha viruses, is E1 and E2. Then you have the virus, which used to be one of the members of this Bunyavirales order, in which GC and GN are the same thing. And for hantaviruses, which are also Bunyaviridae. So they do, although there is no sequence similarity if you compare this, they have the same fold, and the accompanying protein is doing exactly the same thing, masking the fusion loop, which is indicated in orange there, and there is dissociation of this accompanying protein that will trigger the fusion. So, so these heterodimers here are extracted from this other slide. So these were supposed to turn, but there are so many atoms there that my computer gets stuck whenever I make them turn. So you have they're starting wanting to turn, but never mind. So you have these heterodimers that I shown you. I extracted from this structure. So this is the immature particle of a flavivirus on the left. Then the alpha virus, in which you have 240. On the flavivirus, you have 180. For the virus, you have 720 copies of this heterodimer. And then for the hantaviruses, which are pleomorphic particles, they have more of the hand. It depends on the size of the particle, and but they are organized in a similar way, in which the fusion protein in 3 colors makes the lateral contacts to make the lattice, and the accompanying protein makes the center and stabilizes the spike. So, one of the things that we have learned is that these APs is from alpha viruses and from hantaviruses are structural homologs. How do we tell that? So this is what we call a topological diagram, which will start on the end. You see there is a circle marking the end on the red strand. You see in domain A, there is a little circle. I cannot point to this, unfortunately. So if you label the strands from the end terminus by A, B, C, D, you see that you get the same pattern. I can barely read it from here, but it's C, E, B, A, C, F, C. And if you go and look on the alpha viruses on the right, this is E2, which has some insertions before, which is triggered to E3 there. You have exactly the same ordinance, D, B, A, whatever. And this domain is sort of N-terminal to this beta ribbon that we call in blue, and which at the turn will have another domain in yellow. It has to do with exactly the same connectivity. Then you complete that beta ribbon and have yet a third domain with an immunoglobulin structure. So for the chances that 2 proteins that will do something similar, just accompanying, will evolve from 2 different origins to this is extremely likely. That all of the connectivity is maintained exactly. So that the likely explanation is that there was an ancestor that already worked like that, and it's been maintained, although there is no similarity in sequencing when you compare the sequences. And they go to the other families, like the nidovirus, like Crimean Congo hemorrhagic fever virus. You have in red the same motif that we found in hantavirus and in alpha viruses. It has some extra strands than in white there. And on the beta ribbon, the yellow domain has lost some of there. And there is also a cleavage. You make GP38. You say there is a cleavage site indicated there. And then, but you just have the third domain, domain C, at the end. And finally, when you go to viruses, you have the same motifs, except that if you look carefully, you don't find any more this connecting. So apparently, these domains have been exchanged within the same framework that we have here, the beta ribbon and the C-terminal domain. So it's indicating that this accompanying protein is much more exposed to antibodies after variations, and it varies much more. So the fusion protein on MFP, you can really distinguish the fold. It's did not, whereas here they have become quite distantly variant. So just to summarize, the structures of the accompanying protein indicate that the class 2 proteins derive from an ancient AP-MFP accompanying protein, memory fusion protein, heterodimeric fusion machinery, and that also, I don't have the time to go into this, that flaviviruses are the exception, and we found that this exaptation there of a cellular chaperoning from the DNA J protein, from the Golgi, that was being incorporated to the virus particles and plays that role. So this is, see, these kind of things that have been during evolution here. So one last thing I want to say as an introduction is that all of the class 2 viruses we have studied enter cells via receptor-mediated endocytosis, and it is the low pH in the endosomes that trigger this conformational change. So this is, when this uptake into an endosome, the low pH will trigger dissociation. The fusion loop there, this orange star will insert into the membrane that triggers trimerization in an extended intermediate conformation. And then you have domain 3 there in blue, and all my will find the binding position on the side that will lead to this trimer formation and bringing the two membranes together, and plus forming a fusion pore after by going to hemifusion. So that's the mechanism we have. And so what I will focus now is on that particular trimer of hairpins, which is the post-fusion form. And this is what we've found lately, is that the ancestral gamete fusogen HAP2, it has the same fold you see it from Chlamydomonas, from Arabidopsis, and next to the alpha virus ones. So it's the same trimer of hairpins that we have, and you see you have the tree of life on the side, and which you can see the eukaryotes. It is very simplified, but they are emerging on one of the branches of the archaea, and this HAP2 has been found in all of the members of branches that very deeply root into this tree here, sort of suggesting that the LECA was already undergoing sexual reproduction. This is also supported by because they have all of the enzymes involved in meiosis. So, and the protein responsible for the fusion of the two gametes, sperm and ego, is that one. So we've solved it on two members of the HAP2 structure of the plantae, Chlamydomonas and Arabidopsis. They are in the same branch of the plantae. And they had this characteristic trimer of hairpin postfusion conformation, which is this, as I said, the inert lowest energy conformation of the protein adopted after memory fusion. So we know that for viruses, the active protein is this heterodimer that I just shown you, with an accompanying protein that controls. But here, we don't know. We still don't understand, and that's the big question now, understand how these proteins function in this case. So as I say, in contrast to intracellular fusion or virus cell fusion, there is no mechanistic description for the merger of two gametes to make a zygote for any species, the spine being a key event in eukaryotic cycle, and the molecular mechanisms of fertilization is something that's been in the air, but it's nothing really understood at molecular levels here. So the questions that we have now is to understand what controls the fusion activity of HAP2 and what is the trigger. So one key thing will be now to identify the accompanying protein that controls this, if it exists. And we are trying to, in order to try to make an answer, we're looking at members of the different branches. You see excavates contain the trypanosoma, leishmania, and others, which branches really very, very, very deep in the tree. But there's also apicomplexa, and this is also related to the topic of infectious diseases, these membranes, the apicomplexa, contain many multiple parasites, and for all of them, the sexual stage is key for the transmission. So, for instance, in Plasmodium, when it goes into the blood stage, you have gametocytes that are formed. The mosquito will breed it, and they convert into actual gametes that will fuse, and it is the zygote that will cross in the gut of the mosquito in order to infect and to be able to transmit. It will make an oocyst. So there are ways, it's important to understand this for the transmission blocking strategies. And similar for kinetoplastia, trypanosoma is in the tsetse fly, for instance, for trypanosoma brucei. So we are also interested in the hemichordates. That's the amorphia is the branch where we are. But the hemichordates are the closest to the vertebrate for which HAP2 has been identified. In any vertebrates, we don't find, probably this protein may have been exchanged, and we don't have it yet. So if we go to trypanosoma, so we were surprised when we tried to make the same for trypanosoma, and instead of getting that trimer, post-fusion trimer, we got this dimer that is shown here. And Eduardo Vaquero, who did this in the lab, realized that the packing, so there is a molecule behind which is in gray and the front one in colors. The interaction is mediated mainly by a loop. And when in this loop he introduced a deletion on that little loop, and then bingo, we got that post-fusion form, the trimer. So we are already seeing that the trigger to go from pre-fusion to post-fusion appears to be different depending on which branch we're taking in the tree. But the other thing that is very interesting on this particular dimer is that when we compare to, for instance, to the E1 protein, the structure that we got more than 20 years ago from SFB of E1, which is the fusion protein, we got this dimer cross here. And you look now at the reconstructions on the arrow that says top view is the same dimer seen from the top. And that dimer is making the packing between the two adjacent spikes. You see the model in which the trimeric spikes are in different colors. So the dimer that we capture in the crystal structure is this dimer contact in between two spikes there. And then later on, when we did the structure of the hantavirus, we got the same structure of a dimer crossing like that with the fusion loops toward the top. Which we now know is the packing in between spikes, although in this case the spikes are tetramers, and you have in both cases the AP, the accompanying protein in gray or in color, that is making the cementing, forming the spikes. So although the virus particles look very different, there is a basic similar thing. So, and this sort of is postulating that what happens here. There is an accompanying protein in the trypanosoma HAP2, so that this suggests that these proteins will make like a structure will modulate the membrane at the site of membrane fusion that make there. So what I wanted to say is this is an illustration of the power of doing comparative structural studies across related but evolutionary distant systems to generate new hypotheses. And now we are really trying to pull down experiments on HAP2 by using antibodies in order to try to identify this accompanying protein. So the questions is, as I just said, do these AP HAP2 heterodimers make patches with a regular surface lattice at the cell surface? And also how much variety there is across species in the specific control of HAP2-driven membrane fusion reaction. So the one problem we have to do this is that the gametogenesis is only induced in the tsetse, the salivary glands of tsetse flies. But there are new methods that are being developed to induce gametogenesis in tissue culture by some proteins that stimulate that. So we have, as a follow-up, we have immunized mice with our recombinant TB HAP2 and with Brice Rotueux, who is in IP Paris and Institut Pasteur Guinea. we identified that one can recognize HAP2 in gametocytes upon induction of this protein here. And so we now need to upscale in order to so if that was for kinetoplastia, we're looking to the complexa that are in this SAR branch, which is really far away from the excavates. So we express also HAP2 from Plasmodium falciparum for structural studies. We found that the recombinant Plasmodium falciparum HAP2 purifies as a monomer that trimerizes over time, like was the case for Chlamydomonas and Arabidopsis. And we are now immunize alpaca with this recombinant protein and made several VHAs, single chains. How am I doing with time? Should I end? No?
We have 10 minutes.
10 minutes, okay. So to help for the structural determination, but we sort of looking at this up2, we see that if you look at these liposomes, it will float. When we get, it's the trimer that will bind to the liposomes, as Andres and Edward have been working on that. And if you compare to what we were doing like more than 20 years ago on the sembriciforest counterpart, you can see that the arrows are pointing to these trimers, and you see the same thing on the black dots are gold because we're doing tomograms. And so these are class averages in which you see two trimers side by side. What we had found from the sembriciforest was that they also make a lateral arrangement in the post-fusion form at the surface. And so this now, oops, that should have moved. So this is supposed to be a moving. Leah, can you turn it? She said she... I would have needed to click on the image, but maybe someone can do it there. No? That one. Well, never mind. No, this is... On the yellow, there, okay. So you can see the bilayer and the protein sort of bound there. This is very low resolution, and now we're trying to improve the resolution by cryo-EM to understand how these particles... But it turns out that this yellow domain is very flexible with respect to the red, and that's causing us problems. But in addition, the VHHs that we generated, we had a collaboration with Andrew Blackbrough, who has a center in Burkina Faso in which he has children who are infected with Plasmodium falciparum, and he has this test in which he could make the mosquitoes feed on blood from his children, but he adds the VHHs that we gave him. And so he found that, for instance, some, like VHH20 there at the center, the red is the control, and doesn't do anything, whereas others, like VHH4 or 22 or 25, they completely block transmissions at he tried the lowest concentration he tested was 5 micrograms per ml, and that was already too low, so it was completely blocked. So this is one of the uses of these VHHs, and we're also trying to use them to pull down and identify the companion protein for this. So for these VHHs, we're also using them for structural studies. So that's the trimer, and this is VHH46, which did not block transmission. And that, for the monomer, what you find strange here is that we see domain in blue and red at domain 1 and 2. We don't see the yellow domain projecting from it. And the same for the dimer. And here we have a VHH that binds to domain 1 in red. And to make it bigger, we have an antibody specific for the tag of this green VHH in order to make the reconstruction. Yet the domain 1 that we're interested in has the fusion loop. It's mobile, and we don't see any density for that. And just to, I wanted to bring your attention to this, is that on the left, we have VHH46, which maybe reduces a little bit, but it binds to domain 3 and it binds to the trimer. Whereas the one on the right, which is 42, almost at the end of the list, it's very efficient in blocking transmission. So we also did the crystal structure of this VHH22, which is 22 is one of those that block transmission and binds right there. But in order to understand this, it's better to Can you make that turn, please? So this is the AlphaFold model of the HAP2 trimer. Can you please activate there? So I cannot click on. Well, never mind. You see, the AlphaFold model is, the predictions are in blue, are color-coded there. Everything that is light blue is very well predicted. Actually, everything over 70, you can sort of believe on. It has sort of this extra stuff in orange on the side that the program doesn't really know to do, and the fusion loops. This region that we cannot really see because it's mobile in cryo-EM structures. So it does not turn? No. Well, never mind. This is the only reason I wanted to show you this is that because in that model, when we superpose the domain 1 with the antibody, we see that the antibody clashes there and there's no trimer formation. So what the VHH clearly blocks formation of this trimer that is necessary for fusion which sort of supports that the mechanism for virus fusion trimerization is required for HAP2-driven gamete fusion. And the question remains about this potential accompanying protein then. So what I wanted to say is that the apicomplexa is not just Plasmodium. You have Plasmodium somewhere there within them, but you also have Coccidia, Babesia, Theileria. So Plasmodium is in the green at the very top. You have Cryptosporidia. You have the gregarines and you also have Toxoplasma. So there are multiple parasites that are transmitted. And for instance, in the case of Toxoplasma gondii, differentiation occurs only in the cat, in the intestine of the cat. So, and if you could vaccinate cats against HAP2, that will block that and will block zygote formation and cytosis. So you could be in that. And in addition, there are multiple of these members like the Coccidia or Babesia that cause huge losses to the food industry worldwide. And as some for instance, Babesia, they're transmitted by ticks or by other parasites and other directly. And there is a lot of things that could be done. So we started from studying viruses, ended up in eukaryotes, and now seeing that this is a vulnerable step in these eukaryotic parasites that we could be targeting. So just to finish, so I'm giving you here the evolution of life on a different context. So the life started like 4 billion years ago. The Earth birth will be at the center, where they're at a little point, and then the separation of eukaryotes there with the archaea occurred at about 600 million years ago. And you can see they have two distributions. So except for the fungi that apparently have lost it, you can find it all the way and right to below under the vertebrates that come here. And for comparison, I can show the mammals are in the dark brown, so they have syncytins. Syncytins has been also incorporated into the germline of a precursor of mammalians, and it's what makes placenta by inducing formation. But so you can think, so these are these virus exchanges that make evolution happen in a way, and sort of what it's telling you that interaction, genetic interaction between viruses and cells, in all that period previous to the appearance of eukaryotes and the separation of, they have been driven by interactions of viruses with cells and genetic exchanges. And if we are here today, it's because of viruses. And one thing I wanted to plead about viruses, we only study them when they're nasty, and we saw the ones that Charlie told us, HPV, and they can cause really, but they are part of life as a whole. Life evolved in the context of that. And we need to keep tackling those cause pathogenesis, but understanding the whole world of interactions that have focused on. So the genetic exchanges that viruses have made possible have made evolution of life possible on Earth. So let me just acknowledge it. Well, these I told you, many people have participated in these studies, and the most recent ones are on the HAB2, Eduardo Vaquero and Andrés Ferriño in particular, and the core facilities at Pasteur Paris. This is the funding, and thank you for your attention. Sorry.
Thank you very much for the wonderful talk.